The evaluation of genetic risks for non-white populations can be enhanced by a novel approach. The National Cancer Institute and Johns Hopkins Bloomberg School of Public Health have collaborated on a project led by a team of researchers. The team has successfully created a new algorithm for risk-scoring genetics, which can be applied across a diverse range of ancestral populations. The algorithm shows great potential for reducing healthcare disparities in the treatment of major diseases. The use of genetic risk-scoring algorithms is viewed as a promising approach for identifying individuals in high-risk groups who could benefit from preventative interventions for a range of medical conditions and diseases, including heart diseases and cancers. These algorithms rely on extensive genetic research, which has established links between specific DNA variants and elevated or decreased risks of developing certain illnesses. In the realm of genetic studies, the bulk of subjects have been individuals of European descent. As a result, the algorithms used to generate risk assessments have not consistently yielded accurate results in other populations. This discrepancy can be attributed to the genetic variations that exist within different ethnic groups. Today, an article in Nature Genetics outlines a new technique that has been implemented in analyzing genetic data from 23andMe Inc. and similar studies, which collectively involve over 5 million individuals across a variety of ethnic backgrounds. This method has resulted in genetic scores for 13 diverse traits, including health conditions such as depression and coronary artery disease, in 5 distinct ancestry categories: European, African, Latino, East Asian, and South Asian. Additionally, the authors of the paper conducted large-scale simulation studies to verify the effectiveness of the new method. According to Nilanjan Chatterjee, PhD, the senior author of the study and a Bloomberg Distinguished Professor in the Department of Biostatistics at the Bloomberg School, our approach proves effective in narrowing the risk-scoring performance discrepancy for non-European ancestry populations. However, we arrived at the conclusion that the employment of novel methods alone is insufficient to eliminate the gap entirely. We require more significant data sets on these groups to achieve that. Risk-scoring models generated from genetic studies in non-European ancestry populations often lack accuracy due to their limited sample sizes. Consequently, there is a disparity in the performance of risk scoring between populations of European and non-European ancestry, which could potentially contribute to inequities in healthcare. The researchers have introduced a novel approach referred to as CT-SLEB, which integrates multiple AI methodologies such as Bayesian statistical modeling and machine learning. The CT-SLEB model was trained on a wide range of data sources, including the 23andMe database, the Global Lipids Genetics Consortium, the All of Us research program by the National Institutes of Health, and UK Biobank. Upon conducting benchmarking analyses, the research team discovered that the newly devised risk-scoring models, specific to each ancestry, exhibited superior performance among non-European populations. In contrast, the standard polygenic risk score models, which are derived from predominantly European ancestry datasets or smaller non-European ancestry datasets, failed to match the efficacy of these novel models. After conducting research, the experts compared CT-SLEB to several other methods. They discovered that CT-SLEB is especially useful for enhancing genetic risk scores in African ancestry populations, where the accuracy of such scores is typically low. Additionally, the researchers found that CT-SLEB is significantly faster in terms of computational speed compared to its closest competitors. Therefore, it has the potential to analyze a larger quantity of DNA variants across a wider range of populations with ease. According to Chatterjee, the team is presently utilizing more sophisticated techniques that perform even better than before, while still being computationally rapid. The study's results have led him to place a strong emphasis on the importance of developing polygenic risk score models that work equally well across both European ancestry and non-European ancestry populations. He notes that, based on the team's calculations, achieving this goal will necessitate conducting more genome-wide association studies within non-European ancestry populations. According to Chatterjee, many individuals hold the belief that artificial intelligence and machine learning possess the capability to perform miraculous feats. However, without conducting extensive, well-crafted research, algorithms are unlikely to offer much utility. Hao Yu-jung, PhD, who was a doctoral student at the Bloomberg School when the study commenced and now serves as an investigator at the National Cancer Institute, is the lead author of the paper. The development of the new technique and analysis of data were aided by researchers from 23andMe. The CT-SLEB code is accessible via GitHub and can be found in the code availability section of the paper, which includes a link to the CT-SLEB code. The National Institutes of Health, NIH, were responsible for providing funding, specifically K99CA25651301, R00HG012223, 5T32HL007600437, R35CA197449, U19CA203654, R01HL163560, U01HG009088, U01HG012064, R01HG01048001, and U01HG011724. These numbers are exact and should not be abstracted or replaced with synonyms. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.